and at the Zurich School for Applied Sciences and HESO in Valais. She holds a PhD in computer science from the University of Geneva and a master's in philosophy and machine learning from the University of Cambridge. And she focuses on interpretable deep learning methods to support and facilitate knowledge discovery in biomedical research. Maybe for practical reasons, if you have any questions, please put them in the chat. Then I will ask them later, or after the presentation is finished, raise your hand and then you can ask them in person. It's my experience that interrupting speakers during webinars is not very successful, so I'd rather do it this way. So Mara, go ahead.
Wow, thank you for a very nice introduction. I'm really happy actually to be here and to present my work. There's going to be a bit of my PhD thesis work and a bit of my ongoing work and ongoing research. And I decided to give the title like Interpretability of Deep Learning for Microscopy Imaging, and then we will go from like model validation to more exciting areas of knowledge discovery that are something of my current work. And I was planning on introducing myself, but actually, I think the introduction was nice enough, and I think it contained all the key information. Maybe the only couple of things that I can mention are about my funding. So I'm currently funded by two projects. One is the Swiss National Foundation on characterizing colorectal cancer molecular subtypes, and the other one is more on characterizing AI and AI safety for the media and the society, and this is a European project. The work that I will present today is obviously done in collaboration with several researchers. I would like to acknowledge them. So this is not I didn't do it all on my own. It's work that I've been doing with plenty of nice researchers, and I've also been given a lot of feedback by different pathologists at the University of Bern and at the Radboud University Medical Center. So moving on, the overview for today, I think I will structure the talk in this way. So we will start from a very general motivation and a quick discussion about the vision. So about what is my vision and what do I think it's the way we should interpret not only interpretability, but AI in general for digital pathology. And I will quickly go over the state of the art and the state of few of the limitations that I see right now in the state of the art, and I will discuss how I worked during my PhD thesis to actually improve these limitations, starting from improving the explanation understandability and the utility in terms of clinical practice, and then moving towards improving model performance, so using interpretability in a proactive way to actually get better performance of the models. And finally, I will move to my postdoc research right now that is actually using interpretability for science, so to discover things that actually might be useful and that could help us to create hypotheses for further things to test and to verify in the lab. And then we will have obviously time for some final remarks, discussion, and questions. So the motivation that I would like to introduce here is very broad, and it starts actually from a problem that is seen in a wider community, that is the machine learning community. Now, machine learning models are approximations, and they're never exactly true. They're just approximations of the true underlying phenomenon. And to estimate the usefulness and applicability of these approximations is a problem that depends on multiple things. It depends on the context, the task, and the risk that is involved in each application. So we need to always consider the application and the risk that is related to it. There are several proofs that show that deep learning models not always predict outcomes as we would expect. Even if they have impeccable performance on testing data, we might see in a variety of situations performance drops. For example, we just take shifted data and we see performance drops. We can see lack of robustness to adversarial examples, for example, so to this input that are engineered to trick the models. And we also observe the observation of bias and discrimination within the learning procedures. These are all pathologies of deep learning models. And I think that these pathologies are showing us and are screaming and shouting out to us that the evaluation of these models by their sole test performance is actually insufficient, and that we should drive the development towards building new ways of measuring accuracy that can help us characterize not only the model accuracy, but also model reliability and safety. So we need to keep the accuracy, of course, but we want to introduce reliability in the system. Now, at this point, we mentioned interpretability, and I would just like to be able to give a word on interpretability. And the reason why I would like to say a word is because there are many definitions that exist in the field, and these definitions are used by different figures. So there are scientists in the technology, like me, for example, that have a certain interpretation and a certain understanding of the word interpretability. There are people in the cognitive sciences, in sociology, people that work in the legislations that write the policy that then are used at the European level and that are encouraging the use of reliable AI. And these people are different figures. They have a different background, and they interpret the word interpretability in a different way. So technically, what a lawyer would say is that there is no unique definition of interpretability. This is a concept that has to be seen a bit as privacy is generated the same debate. So they see interpretability in the same way, for which there is no unique definition. And my personal take on this is that we actually, at least in the technology and the sciences, and we can try to find an agreement, obviously, between the two fields, but at least for the technology, we can inherit a definition of interpretability from mathematical logic. And in mathematical logic, interpretability is the capability of assigning meaning to an instance by a translation that does not change its original validity. So we are trying to look for a translation of the working principles of an AI systems and of the working principles and of the outcomes of this AI system, and we're trying to find a translation that does not affect the validity of the system, and it is understandable by humans. So we need to create human understandable language. And this is the challenge of the field, essentially. And the vision that I have for clinical applications, for medical, for biomedical applications, is that we need to put the human in the loop. And we need to stop evaluating models by their sole test performance, and we need to build AI systems around the need of physicians. And when I say we need to build AI systems, I mean we need to start from the data collection, the model development, and we need to provide the clinicians tools to not only performance metrics, but really tools to evaluate and understand and create a mental model of what the deep learning model is doing, so that then they can give it feedback to collect better data sets and to build better models. So that's kind of the effort that we should be aiming for. Now, I don't think I need to really introduce to anyone here the context of pathology, but I think that it is a good example of how interpretability is essential in this domain and in this context to guarantee the successful interaction of pathologists with deep learning outcomes. So in these gigantic images, there are some tasks that are quite cumbersome to evaluate manually, and isolated tumor cells and small cell clusters are an example because they occupy a very small portion of the image. And computational models can aid the diagnosis, they can leverage the vast amounts of data, and they can actually help solving these tasks that are a bit more cumbersome for a human pathologist. And I like to report this example at the Beck Lab because they show that actually when pathologists cooperated with the model. So if they used the model on their own, they couldn't beat pathologist performance, if you want. But when they cooperated together, when they relied together on the tool and accepted the help of the tool, they actually reduced the error rate of 85%. Now, this is not a success story because the problem behind this is that there is a leap of faith that is required by pathologists to trust the models entirely. particularly if there are hard cases or cases where making a decision, it's actually complicated. And in the end of the story, the pathologists are the sole people accountable and legally responsible for the diagnosis. So if they decide to rely on an AI model, and this AI model was not safe, was not reliable, was not accountable, and if this is not introduced in the European system that we have now for accountability, then the pathologists and the clinicians are the sole people that are actually responsible for this. So we do need something to essentially let clinicians interact with these models so that they can probe their behavior and build their own understanding of this. Now, the machine learning literature has seen an explosion of papers concerning interpretability, and these papers can be organized and sorted, and they can be structured over different axes. There are some people that try to interpret the data simply by doing dataset exploration. Then there are some people that try to interpret the model by visualizing it to learn patterns and analyzing unit and layer interactions, or analyzing the overall structure of the model, for example, comparing convolutional neural networks to transformers. And then there are other methods that actually became very popular in medical imaging, that are those methods that focus on explaining the prediction, so identifying relevant input features or identifying high-level concepts that can be associated to the model outcome. So these methods are very often post-hoc methods that mean that essentially you don't need to retrain your model. You can just apply them to any trained model that is working on your data, and they preserve performance. So they can be applied without the need for retraining any of your model parameters or changing your model performance. And probably that's why they also largely took the field in healthcare. Now, I think if we look now at the visual explanations that we obtain by applying these models, we actually see that that the current methods do not address the needs of daily practice sufficiently well. For example, if you look at these explanations here obtained by GradCam, that is one of the state-of-the-art methods for explaining post-hoc models, you can see that the method is pointing to some nuclei that appear maybe enlarged or that appear with a weird texture, and that the contours are very neat. So the model is looking at these nuclei to make the decision and to classify these patches as tumorous patches. The problem is that it also generates heat maps that are very diffuse and that can create some confusion in explaining what the model is looking at. And the similar results are obtained with another state-of-the-art method that provides very dizzy and fuzzy contours, and they don't clarify whether the model is looking at the nuclei or at the context of the background, for example, to make the prediction. So how can we improve actually the understandability of these explanations? How can we make them more effective and easier to interpret and understand in the first place? So ideally, I think what we would like to have is something like this, where we actually point out to the instance that is mostly being picked up by the model. And by instance here, I want to attach a semantic meaning to this instance. So in these images, we have nuclei that have been segmented manually, for example. If we don't want to use manual segmentation, we know that there are ways to generate these segmentations. So we can isolate with more or less accurately what are the instances that there are in these input images. And here I'm focusing on nuclei, but there are also other ways that focus on lower level structures, if you want, like stroma or mucosa, depending on the tissue. And while this is the result of state-of-the-art models, this is what I proposed in a paper that's called Sharp Line that was published at MICAI. And here I'm trying to use these segmentations that we know how to obtain to actually generate explanations that have a semantic meaning and that isolate instances in the inputs. And so here you can see that actually the model is picking up what it seems to me to be the largest nuclei in the image. So also this one is picking up the nuclei in the image while is neglecting the background or maybe focusing only on some parts of the background. And we actually wanted to evaluate these methods by involving experts. So we asked them which explanation. We designed this interactive tool where they could navigate the slide and navigate the predictions of the model. And we asked them which explanation method among 4 they found the easiest to understand. And so far, the majority of the experts is actually giving us, telling us that sharp-line is indeed easier and more immediate to understand. But the interesting thing is that actually pointed us to further clarifications of what are the requirements that these interpretability methods should actually meet up to. And the biggest clarification that emerges for this study is what I call the loss in translation phenomenon. So the experts would expect explanations actually at the level of the entire image, or they would expect explanations that could zoom in with the image or could navigate with them, and that might overlay or not, depending on on their preference on the entire image. Now, the problem with this scalable version of the explanations is that, at least for this model, it is not possible to generate explanations in this way because they would not mirror how the model works, because the model that is working behind this tool is actually only looking at individual patches of tissue regions. And so we need to create new development and to create new methods that can actually further address this requirement. But overall, I kind of think that we have the confirmation that we should move towards sharper and more immediate explanations that can meet the requirements of clinical practice. Now, another problem that I would like to mention is that even if we do find high activated regions with these heat maps, with these visualizations, might be GradCam, Lime, or SharpLime, it is difficult to abstract what they actually represent in terms of clinical features. And here is where the idea came from with concept attribution methods and the idea to generate user-friendly explanations that are expressed in terms of clinical concepts such as nuclei morphology. So this image here is adapted from a pathology manual, and there are clinical features that are explained in relation to the tumor grade associated to breast cancer. And these features are the degree of tubular formation, the nuclear pleomorphism, and the mitotic rate. And the grade increases, for example, if there is a lower degree of tubular formation, if the nuclei appear larger, have vesicular appearance, high chromatism, and obviously if the mitotic count is very high. So starting from this, I developed these regression concept vectors as an extension of a method that existed already in the machine learning domain that was proposing this idea of analyzing the models with concepts. And the approach is quite straightforward. So we use, again, the segmentation, might this be manual or automatic, of the input regions in the areas that have semantic meaning, for example, annotations of the nuclei. And from these segmentations, we can extract handcrafted features that represent the concepts that we wanted to analyze. In the case of nuclei morphology, for example, we could look at texture, size, and shape of the regions. And then we pass these images through the model, and we take the internal activations of these models at a given bottleneck layer. And the idea is then to see whether we can actually learn in this bottleneck network with a simple linear regression these measures that we extracted at the beginning of nuclei area, texture, and shape. So ideally, what we're trying to look for is a regression that tells us the direction of change in the learned representation of a given concept. And we can use this direction to actually see how this direction impacts the gradients of our model so that we would know the sensitivity of the model output to a given concept. And so this is something that I have applied to the same problem of classifying patches of containing tumor from patches that did not contain tumor. And this is one of the outcomes that I obtained for this model, where we see that nuclear morphology actually appears to be a relevant feature that is picked up by the model, because we do see that there are features of correlation and contrast that strongly change the model output, so that have a large impact on the gradients. And the nice thing is that we can obtain global explanations at the entire model level, so we can compute the explanations on the entire input data set, but we can also compute them individually for each patch. And so here you can see that this patch is taken from a tumorous tissue. The prediction is tumorous, and we do see that it is the dissimilarity and the contrast in this image, that are visual features representing texture, that are pushing the gradients, so are pushing the model to predict that this patch is tumorous. In this case, there is not much content in the image, and we see that the values are actually much larger, meaning that if there was an increase in the similarity and contrast, then this would strongly affect the gradients. So you see a bit how each patch has a different explanation, and how these explanations actually create a coherent picture of what the model is picking up. Now, can we use this somehow to actually get better models and to improve the performance that we have right now? So this is one of the things that I wanted to focus on, and I thought of this. So most of the models that were used and that are still used in the field reuse features that are trained on natural images. And so they take very often people take an ImageNet pre-trained model, and they then either do fine-tuning, or they add to the feature extraction, they add further layers to then classify these histopathology images. Now, there is a fundamental problem with this, because if you see here, the ImageNet collection depicts thousands of objects. And for these objects, the observation viewpoint from which the images were taken is unknown. And so here you can see that this is an image of an albatross, and the observation viewpoint was very different in these two. Here we're very close, here we're very far. But these two images are still belonging to the same class, that is the albatross category, because they're both representing albatrosses. Now, I feel that in microscopy images, things are different, because the observation viewpoint is fixed, and the object scale has actually a corresponding physical measure that has a meaning. So if we were looking at the entire whole slide image and measuring the diameter of this tumorous region, then we would know that this actually diameter corresponds to a physical measure, and that from this we can derive a diagnosis. So you see that there is some real meaning associated to scale in these images, whereas here scale is actually doesn't matter. So the idea was to use an ImageNet pre-trained model and to use regression concept vectors to see how scale is learned in these models, and actually how it is incorporated at each layer. So we can apply what I explained before at each layer, and we can quantify how scale information is actually to what point we can predict scale information. And we see that it peaks at some point, and then the model starts learning this invariance to scale, because it actually is assigning objects at different scales to the same class. And it's actually this decrease in the scale information content in the embeddings that we want to remove. So we can actually prune those layers that are giving us the scale invariance, and we can use the pruned model to transfer to histopathology tasks where we know that the scale is relevant. And we tested this on the regression of RH nuclei size. So we chose this task of identifying whether the images were at 40x or 20x, and we see a massive improvement in performance when we actually use the prune layers. So this is, I think, a very nice and easy example that we can use these methods to actually make hypotheses of what we think that should be actually incorporated in the representations, and how we can obtain that and enforce that in the models that then we use for our final application. Now, going to the last part for this talk, I will just give a quick overview of some of my fresh work on using interpretability actually for discovering knowledge and for creating hypotheses that then could be used for science. So the motivation behind this is that actually we only are scratching the surface. There are many things that deep learning models capture, but that we don't know. And there are examples that show us this. For example, CNNs can predict the gender, smoking habits, and risk of cardiovascular disease from eye fundus imaging. And I recently talked to an ophthalmologist, and I said, I'm sorry, I really need to ask you, but could you predict these features with very high accuracy from eye fundus images, given your experience and given the number of people and patients that you attended? And he said that that it's not possible for the human eye, for the naked eye, to actually predict these features, whereas CNNs seem actually to be doing this quite accurately. And another thing that is very interesting to me is that patterns of gene mutations can actually be predicted from human tissue microscopy. So there is this paper where they take all the whole slide images in TCGA that have associated transcriptomics of gene expression, and they see that they can actually predict gene mutations, DNA mutations, and so on and so forth. So the idea is this: can we use actually interpretability to discover what relationships are picked up by these models? Because we actually are not aware of what relationships are being used to make this prediction. So maybe we could try to understand what these are and make hypotheses about what the features picked up by these models look like, and see whether we can go anywhere with that. So the application case here is this example of colorectal adenocarcinoma, and the reasoning, like, why should we be doing this and why would this be useful? It's because actually 80% of colon cancer is adenocarcinoma, and the diagnosis of adenocarcinoma is quite visible already at the macroscopy level. The interesting thing is that there are different ways that adenocarcinoma can show up, right? So if we look then at the microscopy, we start seeing all the differences between the tissue, and the diagnosis and the prognosis differ depending on how the tissue looks like. But there is even further to this, that is that molecular subtypes, so different types of transcriptomics, can differentiate the prognosis of the patients that have colorectal adenocarcinoma. The problem is that RNA sequencing is costly, takes time, and is rarely integrated in the clinics, and is not directly available to oncologists. There is previous work that tries to predict gene mutations, so this is motivating. And so the idea is really, can we try to learn any relationships between the microscopy and the molecular patterns? So the way I'm proposing to do this is to the so-called semi-interpretable modeling. So essentially benefiting from attention-based mechanisms, such as the one in attention-based multiple distance learning models, so that we could use attention as a means to interpret the model. And the idea here is to transform the attention-based multiple instance learning proposed by Ilse into a regression problem, so that we are actually learning a single continuous label that is representing the gene expression from a bag of input patches that represent all the patients, right? So we have for each patient, we have a bag of patches that comes from the tissue that was not sequenced. So we have our initial tissue, this part with sequence, we obtain the RNA sequencing, the bulk RNA sequencing for this part, and we take whatever was taken aside and used for generating digital slides, and we split it into patches. We put all of it into a bag, and we say we should use an attention-based multiple instance learning model to actually try to predict what were the values of some of the genes that we think might have a correlation, or might give us some information about the patient subtype, for example, for colorectal cancer. And this is quite a hard task for a pathologist, but we do see not random predictions. So if we compare the performance of our models to random guessing, we do see that we are actually capturing some signal. And what's even more exciting is that we used single cell data to actually validate the patterns that we learned from this model. But I will get back to this bit by bit. So just to quickly analyze the performance of the model. So across all genes that we analyzed, that was a pool of 47 or 45 genes, if I remember correctly, our model obtains a reduction of an overall reduction of 10% in the median absolute percentage error compared to the baseline, because there is a baseline model that wasn't using attention and that was actually predicting gene expression, but we do decrease the error made by these models significantly. And as I was saying, compared to random, we are much, much lower than that. So we are picking some signal. Then there are obviously some genes that are not too well learned, and there are other genes that are learned very well. So we still need to look into how to select genes so that we know that there is likely to be some change in the morphological patterns to be expected due to the overexpression or underexpression of those genes. And now this trainable attention mechanisms provides us some transparency on actually how the model filters the information in the whole slide image. And salient regions can be identified without the need of any other post-hoc analysis or explicit localization modules. Now, there's been some debate about how attention-based explanations should be used. And it seems that the correct way is actually to generate multiple models and then to ensemble them together. So to ensemble these patch-wise attentions so that we would have a robust estimate for the total model attention. And the reason behind this is that there could be simply multiple explanations that are valid. And that also happens in real life, right? There could be multiple explanations for the same thing that are still valid. And these are some of the results of some of the visualizations of the attentions that I could propose. So here there are two different genes, and these are the attention. So the high attention regions for this slide. So this patient had top and bottom sections that were taken off from the tissue that was used for the sequencing, and as you can see, these are the predictions of the model. So we can kind of spatialize this gene expression from bulk to spatial gene expression predictions. And we obviously need to take into account that there is some degree of error to that, but the attention helps us further reduce that, because we know that the attention is actually playing a role in then giving the final bulk prediction value. And another interesting thing is when we look at multiple genes, we actually see that the attention focuses on same regions for some genes and on different regions for other genes. And when we use the single cell data, we actually realize that the attention is focusing on different regions for non-co-expressed genes. Another thing we can do is we can actually look at attention patterns, right? So for each gene, we can retrieve the patches that had the highest attention for each degree, essentially, of gene expression from low to high. And I'm not an expert in this, so I cannot really comment on these visualizations that I achieved. But what I can say is that, to me, these patches look very different from these patches. So the patches that I retrieved for one of the genes look very different for the patches that I retrieved for another gene. But more quantitatively, essentially, what we can see is that if we look at the so obviously it's hard to evaluate these visualizations, and one thing we can do is to test that expectations are verified. So if we have any expectations on the variability of tissue morphology related to abnormal expression in RNA sequencing, then this should be captured by the attention mechanism that we have trained. And the single cell data can further help us with validation. So what I was saying is that if we look at genes that are actually don't seem to be co-expressed in single cell, then we don't see any relevant correlation in patches. So the attention patterns might be do very different things. But when we go to genes that are co-expressed, we see that the attention the attention heat maps start having quite an interesting degree of correlation. So this is telling us that genes that are co-expressed in single cell data, so that are expected to be co-expressed in similar regions, are also matching what our attention mechanism is showing. So the attention mechanism is showing our expectations in terms of ground truth. And so just to conclude, I think that this is a very exciting time. I mean, there is so much that we don't know and we don't understand yet, but we know that we can use machine learning and interpretability to both support physicians on, so in daily practices, the requirements are different, right? We can try to support for unsure predictions, for hard cases, for daily practices that are cumbersome, that require lots of time and include fatigue. We can create some algorithms that really can support the daily practice of physicians. But we can also try to discover new and useful knowledge and formulate hypotheses about this. Obviously, the challenges are many, and this is a long way to go through. I think interpretability needs also to be linked for everyday usage. It needs to be linked to model uncertainty, to fairness, to safety concerns. And for this, we need actually to link communities. We need to link machine learning researchers that work mostly on model development on synthetic data or on simpler data sets. We need to raise their interest to the difficulties that we face on medical imaging data sets. And for this, we have, like I just wanted to mention, we are starting a community that is called Shifts Community, a Shifts project. That is one attempt of raising awareness to the problems related to uncertainty estimations and model robustness under distributional shift, which is something that I haven't talked about, but it is also observed very often in digital pathology due to the heterogeneity, essentially, of the slides and of the different parameters that are used at different institutional centers. And there are also open questions. I mean, how do we actually integrate the explanations in clinical routines is something that is not clear yet. What kind of information might actually be useful and what information might be misleading or even redundant is something that has not clearly been assessed yet. So how do we select then the useful information? And in the case of discovery, actually, how do we assess, verify, and test the discovered patterns? It's also something that needs more research. So here there is a list of references. I probably can share the slides so that you can have a look at this later. But yeah, I would be happy to take any questions. And I mean, I hope you found my talk interesting. Thanks.
Doesn't work well virtually, but I tried. Thank you for the very nice talk, Mara. If there are any questions, I see at least 2 raised hands, and I'll just give the floor in order that the hands are raised. So I have one question from Brandon Gallus, so you can unmute yourself and ask your question. It sounds like your audio is not working. It looks like your audio is not working, Brandon. We cannot hear you. The question is now in the chat. I will read it for the people who don't have access to the chat. I'm not a model development expert. I evaluate models in the regulatory space. Industry wants to show explainable AI outputs to the end user. This requires validation of the explainable AI outputs. At the end of your discussion of regression concept factors, you went right to model optimization. And when I think of an optimization loop, performance assessment is one of the steps. Is there any way or work on either validating explainable outputs or performance assessment of the explainable outputs?
Yeah, I mean, that's touching a good point, right? The evaluating explanations is it's really hard. It's challenging because there is a subjective component related to this, and I haven't talked about what understanding something means, because it's related to cognitive psychology. It's related to how each of us actually modules and captures information and understands information. So there are initiatives on benchmarking explainability. I have to mind at least, there is at least one that comes up to mind, and I can post it in the chat. It doesn't have, I don't think I have seen any medical imaging data sets on that. So there are, the problem is that right now people are really focused on tabular data, on data sets with less features, on natural imaging problems. So there is, the efforts are still related to those data sets. But I do see them expanding in the future and growing, and it's good that there is some activity in this sense. I think at least for what I've been working on, one of the things was trying to assess the reliability of the explanations. So making sure that we actually always obtain the same explanation if we're using at least a post-hoc model, which is not the case for some explainability methods that there are out there. The second thing I think is that we really need the human in the loop, or in this case, the clinician in the loop. So we need to create these interfaces, these interactive interfaces that can help us with data collection so that we can better understand what are the requirements. Because one of the things is the reliability of explainability outputs. That's one problem. And the other thing is also the utility that these explanations might have at all in clinical context, because what, as a machine learning expert, I might think is relevant, maybe it's relevant for model debugging, but it's not relevant in the clinical practice.
Thank you, Mara, for that very extensive answer. We'll move on to the next question from Somfai Gabor.
Hi, everybody, and Mara, congratulations on your talk. As I told you earlier, I was not able to attend fully because I had clinics, which I was supervising, but I found your slides at least very, very interesting. I don't know how many clinicians we have in the round. I myself am an ophthalmologist and very much interested in the clinical use and usability of AI. And I believe, this would be really fascinating to look at the language, how these AI outputs talk to us. So this is what you also touched upon, which of the interpretation more fancied by a pathologist, whether it's the raw one, it's the smooth one. But not only that should be taken into account. One issue that comes with AI in the clinics is the issue of quality. How would AI raise the quality of clinical decisions? This is something I would also love to look at, how you can draw the attention of the clinician to given details within a finding, let it be a retinal finding or anything else. So I would be curious how you see that from the computational point of view. That's one remark and question. So does it really have the option of and how to enhance quality? And the other question is, or remark is, actually how to validate, how can you validate the output of these, or how they speak these AI algorithms? What do you mean by that validation? How can you make a repeatable or somehow objective evaluation of that?
So, okay, so to answer your first question, that was about how do we measure if using interpretability and also deep learning in general, so AI in clinical practice actually improves the quality of the diagnosis and of the outcomes. I think this goes back a bit to my introductory slide in the context of pathology. So this is an example where pathologists supported playing together actually with this AI mechanism that was identifying region of interest, reduced the error rate by 80% or something like that. So I think that we need user studies. I think, yeah, the answer to your question is user studies. And I cannot give it to you because I haven't done them yet. And I think that there aren't enough yet. So we need user studies. We need to actually create a loop and create an interface that we can use to test the algorithms together with clinicians and to test the explanations.
Mm-hmm.
To go through the second point, to your second question concerning the reliability of the explanations, I haven't put this in the slides, but actually one of the motivations behind Sharp Lime, this method that I've developed, is that when you use Lime with different initialization seeds, so I'm going very down deep in the technical details, but you might change one parameter in your code and you might obtain a different explanation. Mm-hmm. And why this is not surprising when you look at the math and how this method was conceived, it might be quite puzzling and not very reassuring to have something like this in the context of medical imaging, because you do want to actually have consistent explanations that do not change if I change one hyperparameter, and also that that show sensitivity to some input parameters change, right? So there is another paper where they have been tricking the model. For example, they have been randomizing the model weights so that you have a model that is just predicting random stuff, and you still have the same explanation. That's also what we don't want. We want our explanation to change with the input content and with the output that is associated to the input content. So that's why I think it should be linked to uncertainty, it should be linked to outlier and anomalies detections, and all of this needs to build a package that is a bit heavier, that is maybe reliable or safe AI if you want, so that we are able to bring this margin and apply AI with safety.
By the way, there's just as an ophthalmologist, a short remark. I've just seen a very fascinating review from Eric Topol on multimodal AI, so that you can combine multimodal imaging or multimodal information coming from multiple channels that could even raise the interpretability, maybe, or it can somehow tweak the system into a direction that is better understandable for the clinicians. Thank you, Mara. Very thought-provoking talk.
I see that there is another question from Klaus.
Hi, everyone. Hi, Mara. Really nice talk. It was,
thanks.
Yeah, so many really necessary insights for the community there. And I agree with also all your answers to these questions, which I think is a very interesting discussion. I just wanted to drill down a bit further on, I fully agree with you that we need user studies with clinicians in order to really understand what works and not. And I also agree with the type of reliability studies you were talking about in terms of understanding whether explainability methods work or not. But to Brandon's question, I'm a bit hesitant whether we ever will be able to evaluate and validate the explanation as such. To me, it's such a close and tight interplay between the user and the model, and it's so easy to fall into one of the pits where we over rely on AI or under rely the AI, and we think that we have some nice concept that we recognize, but it's actually just a simplification of what the AI is picking up. So we don't really understand, whereas we think that we understand. So in my advice to Brandon and the FDA team would really be to just do the user studies and see what works in terms of the joint prediction and efficiency capacity of the AI and clinician team. Would you?
I do agree. I agree with you. I mean, I agree with you. And if I can add on top of that, I think that we need to collaborate with social sciences to design the user studies, because there are some things that go beyond our capacities. And I think if we don't start I think it's an effort that needs to be done by multiple communities at the same time. And we do need to evaluate the joint result, but we do need people that have different types of expertises and backgrounds to design the user studies so that they are complete and they could analyze all the different possibilities. So yes, indeed, there is the risk of confirmation bias that is really high, and we need to hamper it down as much as possible when doing the user studies.
Thanks a lot.
And just, sorry, just to further add, I think that actually the way I see it, and it's that the postdoc explanation methods have seen really a huge and rapid increase because we've moved from handcrafting features to very good performance, but it's due to what? So then we have a method to understand what's going on, but now that we know what's going on, we actually should try to move forward and try to introduce interpretability in the constraints of our optimization. Or if not interpretability, it should be reliability, safety, fairness, all of those, robustness, all of those parameters should be added to the training directly.
Okay, so there's a comment from Brandon. My colleagues and I don't have the capacity, resources, and data to do the work. The community needs to address the open question. I think that's a very nice call to action to maybe wrap up the webinar with. If there are any, unless there are any other questions. No, then I think we should thank Mara again, and I'd like to give the last word to you if you have any final comments or thoughts you want to leave us with.
Thanks. Well, I mean, I think I agree with Brendan that that's what we're trying to do, right? So this Shift initiative is trying to bridge communities, take machine learning engineers and brought their attention to the problems that we have right now, that is actually data scarcity, that is lack of correct labels and lack of labels in general, and uncertain, like domain shifts and all of that. We need to raise their awareness and we need their help because they have better tools, and only joining forces we will probably, so we are trying. It's like, there is light. That's my final remark, I think.
Thank you, Mara. Thank you all for attending today again. If you have colleagues who wish to watch this webinar later, you can point them to the SharePoint where the recording will be uploaded. Also for the people who want to watch this great talk again.
Thank you very much.
See you next time.
Bye. Bye, everyone.
Upload jij de recording?